National Symposium.
Thank you very much. It was my pleasure.
And last but not least, this is awarded to our lovely moderator today, Mrs. Adriani Sunudi. Please kindly receive the award. And now we are entering the documentation session. So for all of the speakers, moderators, and our respective participants, we would like to ask you to open your cameras if there are any hindrance, or yes, if you would kindly open your camera as we enter the documentation session. Alright, I'm seeing a lot of cameras as of now. Maybe I'll take the photo in about 10-20 seconds. So please, if you're about to open your camera, please do it now. Okay, since I've seen a lot of faces here, I would like to start the documentation session. So for the first slide, on my count, 1, 2, 3, cheese. Okay, maybe once more for the first slide. 1, 2, 3, cheese. Okay, and now for the second slide, I would like for all of us to stay on the screen, since maybe you are placed in the second slide. Okay, on my count, 1, 2, 3, cheese. Okay, I think that's it for the documentation session.
The committee will also send gifts to your respective addresses, so please kindly wait for the gift to arrive. Once again, thank you for sharing the priceless experience in this symposium, as we will proceed to the next agenda. Ladies and gentlemen, we will now proceed to poster presentation, which will be showed in the form of videos. The poster presentation session will be moderated by Dr. Feni Sandita. Before we start, I will read his CV first. Dr. Muhammad Fedi Alfiani Sandita is a senior lecturer in IPB University. He finished his bachelor degree in Marine Fishery, IPB University, and get his master degree of science in tropical coastal zone management at Newcastle upon Tyne University, United Kingdom in the year 1990. And then he got his PhD in marine ecology at James Cook University, Australia in the year 2000. He has experience in development of community-based fisheries in 6 locations in Indonesia, and then Mitra Bahari training, and marine protected area management plan training. Since it is presented in the form of video, if there are any questions, please type your question in the chat box below. And for the author to remain online as to answer the respective question to the poster presented. Ladies and gentlemen, welcome to the poster presentation section.
Thank you very much, Sarah and Glenn. Now it is a session for the poster presentation on the second day of the symposium. First, I would like to invite Ms. Anna Aryasari, Ms. Andini Anabila Putri, and Mr. D.G. Herbawa to be ready on the screen. And before that, we would like to prepare the poster presentation that will be put on the screen by the committee. So to the committee, please start the presentation of the poster, and I request all the authors be prepared, for the question. Okay, to the audience in the Zoom,
warahmatullahi wabarakatuh.
thank you to the audience. Please observe the video. Yeah, thank you.
Assalamualaikum warahmatullahi wabarakatuh. Good morning, ladies and gentlemen. My name is Anaria Sari. I am from Marine Science Study Program, Faculty of Agriculture, University of Bengkulu, Indonesia. I would like to present our research about spatial distribution of benthic habitat mapping using PlanetScope image in shallow water of Kahiapu Village, Enggano Island. The lack of information on the benthic habitat in the shallow waters of Kahiapu can be monitored using PlanetScope image, which can provide and extract the daily data and high spatial resolution image. This research aimed to map the benthic habitat spatial distribution using PlanetScope image in the shallow waters of Kahiapu. Lane correction and water column correction were applied to the PlanetScope image. The principal component analysis transformation was also applied to the corrected level band, clean corrected level band, and depth invariant index corrected level band. A field survey was conducted using an underwater photo transect to collect the benthic habitat actual data. Model samples were used to train the maximum likelihood classification algorithm. Validation samples were also used to validate the benthic habitat map. This research showed that the PlanetScope image using PCA transformation produced 40.57% to 53.90% of overall accuracy from the each bin input to produce benthic habitat map. The depth invariant index PC band produced the highest accuracy, while the clean PC band produced the lowest accuracy for benthic habitat map. Seagrass is well classified from the clean PC band and depth invariant index PC band, while macroalgae is challenging to map and can be only produced from the clean PC band and depth invariant index PC band. The spatial distribution of benthic habitat showed that the seagrass beds and substrate are dominant, while macroalgae rarely live in the shallow water of Kahiapu Village. To conclude, PlanetScope consistently produced benthic habitat maps using major level classification and maximum likelihood transformation algorithm, which determine overall accuracy from 40.57% to 53.95%. Okay, that's all about our research. Thank you very much, and wassalamualaikum warahmatullahi wabarakatuh.
Ladies and gentlemen, I will say thank you for opportunity so I can be here with you and I can present my research that I have done before. Let me introduce first. My name is Andi Nabila Putri. I am bachelor student of aquatic product technology in IPB University. Today I will present about characteristic of tilapia scale, gelatin, and its application on surimi. So the introduction. Surimi is one of fish product that has good impact to Indonesia economy, and surimi is a main fish that used for make advanced food product like fish cake, chikua, and kamaboko. Surimi commonly produced by fish that has low economic value and white meat like greasy fish. Surimi that has good quality also has good gelatin ability. Gelatin itself like process that can support a good texture of food product. Usually people improve the cycle of washing meat fish, but it lot of cost, and excessive washing can remove the important protein like myofibril in surimi. So now people use food additive like carrageenan, tapioca flour, salt, gelatin, and all. But some of them can influence the flavor and aroma. Gelatin also limited use because the common raw material for production is from pig that not everyone can use of this. Research now show the gelatin can be made from fish starch like scale fish. So from this research, we see the characteristic gelatin from tilapia scale and application gelatin to surimi curacy, also the characteristic gel surimi. The method we use first is production gelatin. Then we see the characteristic of gelatin tilapia scale. Gelatin then add to surimi with this concentration, then mix two of them with food processor. The surimi was formed into tube shape and heating in 40 degrees Celsius and 90 degrees Celsius, then surimi gel or kamaboko is ready. The result showed the gelatin of tilapia scale has quality that still appropriate with PSN or standard in Indonesia. The yield of gelatin is good. Gel strength higher and most of parameter has similar value with literature Lamila 2022. The characteristic of surimi gel, the adding gelatin of tilapia scale, also has hardness and chewiness that significantly improve as higher concentration gelatin the adding to surimi. Springiness and cohesiveness has similar value. Ability bending water in surimi also getting better. And the whiteness or the color of the surimi gel is getting lower. Maybe it causing by the color of gelatin that we use is a little bit yellowish. The conclusion, tilapia scale can use for the raw material gelatin that has good yield and gel strength. Also, characteristic is appropriate with standard BSN, and the addition gelatin to surimi also has significant effect on the hardness, chewiness, and whiteness. Gelatin with 0.6% be the best treatment on this research. So this is the end of my presentation. Thank you for the attention.
The operator, please display the presentation of poster from Mr. DG
Everyone.
Harbowo. Yeah.
Throughout the session, we would love to share our study about discovering biographical land bridge between Bali, Nusa Penida, and Lombok as the possible dispersal pathway for terrestrial fauna during the Pleistocene glacial periods. My name is Lea Christy, and my colleague is Dani Gatot Harbowo, and we are from ITB. I'm honored to stand before you today to present the findings of our research on the prehistoric landscapes of the Lesser Sunda Islands. We embarked on a study to uncover and understand the geological and biogeographical history of this region, especially correlated to the fauna dispersal and occurred during the Pleistocene glacial periods. Our exploration began with examining numerous paleontological findings, which served as valuable markers of prehistoric life in Lesser Sunda, such as Elephantidae and Stegodon. But did they reach this region? Are they swimming together with their herd? Or are they hidden land bridge that connected the areas in the past time? We are interested to discover the land bridge between Bali and Lombok, but which part of Bali could be the land bridge? At the northern part from Karangasem, or the southern part via Nusa Penida? Focusing our attention on Nusa Penida, we hypothesized the existence of a biogeographical land bridge connecting this island to Bali and Lombok during the LGM. To substantiate this hypothesis, we reconstructed the bathymetric conditions corresponding to the global sea level drop during the glacial periods. This crucial step allowed us to visualize the ancient landscapes that once connected these islands. Furthermore, employing advanced methods, and we rematched the global sea level change data and the bathymetry data from the Indonesian Geospatial Information Agency. By that data, we reconstruct the bathymetry based on it as well. It also followed by conducting in-depth analysis of the Burger gravity anomaly. This examination provides us with notable data, enabling us to estimate the lithological and the landscape of the land bridge. The results of our analysis were nothing short of remarkable. During glacial periods dating back to at least 800,000 years ago, evidence suggests the existence of the land bridge on the western side of Nusa Penida. Directly linking it to Nusa Penida in Bali, the lithological composition is identified as a part of the limestone formation. On the eastern side, a submerged land bridge was revealed. Submerged 225 meters below the recent sea level, possibly comparable to the volcanoclastic to fluvial formation. Yet, while glacial periods end, the land bridge are submerged and triggered by an intensive erosion caused by the relentless Indonesian Trout Flow (ITF). currents in the Lombok Strait, leaving behind enduring imprint of the past in modern. Result of the study show that there is no indication of a land bridge in the northern part of Lombok Strait via Karangasem. The bathymetry of this part is more than 1,000 meter deep, even so minus 225 meter sea level drop occurs. But there are several land mass emerging when the sea level drops to minus 125 meters the southern part of Lombok Strait. It is more intensive and broader and connects Bali and Lombok directly via Nusa Penida, while the sea level drops at minus 225 meters at the Pleistocene glacial period. In conclusion, the southern part of Lombok Strait is possibly part of the existing land bridge connecting Bali and Lombok via Nusa Penida. At the same time, the maximum sea level drop at Pleistocene, our research not only illuminates the rich tapestry of the prehistoric life in the Lesser Sunda, but also challenges our understanding of biogeographical dispersal pathways. The discovery of this land bridge crossing the Wallace Line expands our knowledge of the interconnectedness of ecosystems in this region. As we reflect on these findings, let us recognize the importance of continued exploration and scientific inquiry. For it's through such endeavors that we uncover the secret of our planet's history. Thank you for your attention, and I'm now open to any question you may have.
Thank you very much, the operators. Now I would like to invite Miss Anna Ariasari and Miss Leah and Miss Alina Adela Putri on the screen. So, okay, the committee, please notify me if there are some questions or comments for the authors. Okay, all these repeat questions are very interesting. Yeah, they are about the result of the satellite image analysis for the basic mapping, and then on the improvement of the food process quality, and then also on the question whether there is a dispersal pathway for terrestrial fauna in the past. Sorry, okay, the operators mentioned that there is another poster to be presented. Okay, please speak a bit, Mr. Operator.
Assalamualaikum warahmatullahi wabarakatuh. Good afternoon. My name is Nurlaila Ervina Herliani. Allow me to explain my research and theme about effect of solvents and raw material forms on antioxidant activity of green seaweed Burgesenia forbesi. Human body needs antioxidant intake from outside because constantly exposed to free radicals from various sources. Seaweed has been known for decades to have health benefits, including as an antioxidant. Burgesenia forbesi is a seaweed species that is abundantly found in Bengkulu but is underutilized. Previous research has proven that B. forbesi has antioxidant activity. Antioxidant activity of seaweed can vary due to their extraction method, such as solvent type and raw material form. The purpose of this study was to assess the antioxidant activity of B. forbesi extract collected from Teluk Sepang beach in Bengkulu under different solvents and raw material forms. This research was carried out in 4 steps. First, seaweed sampling. Second, sample preparation. There are 2 sample treatments, namely fresh and dry. Third, extraction. Seaweed has been macerated into solvents, ethanol and methanol. Fourth, antioxidant activity assay using DPPH. The results show the dry seaweed ethanol extract produced the highest extraction yield. Since fresh seaweed has high water content, only a small amount of bioactive component can be extracted. Meanwhile, dried seaweed has more bioactive component than fresh seaweed of the same weight, resulting in increased bioactive component extraction. The polarity of the solvent can influence the extraction yield. According to Wu et al. 2023, seaweed bioactive component will dissolve more easily in solvent with the same polarity. The IC50 of B4BC extract is classified as intermediate to weakest antioxidant and only influenced by variation of the raw material form. Dry raw material have a lower IC50 than fresh raw material, so that dry seaweed had better antioxidant activity. This study proved that B4BC has the potential to be developed as an alternative natural antioxidant. Thank you for your attention. Waalaikumsalam warahmatullahi wabarakatuh.
Yeah. Dear audience, ladies and gentlemen, it is time for us to discuss the substance of the presentation. So I would like to have your comments or question, and I would like to request all the authors to be ready or prepared to respond to the question. And to the committee members, please help me to notify the questions or comments from the audience. We have 4 posters just presented now. Any comments? Okay, probably if there is no such words or sentence, I would like to invite you to give reaction for this session while waiting for the comments. Since time is very limited, I think I would like to invite each presenter to directly make a statement or whatever in this occasion. First, I would like to invite Ms. Anna Aryasari. Please, Ms. Anna.
Okay. Can you hear my voice?
Yes, it's very clear.
Okay, everyone. Our research about spatial distribution of benthic habitat using the planescope image. This is the basic research that we conducted in Enggano Island. Because in Enggano Island is very difficult and has the limited accessibility for mapping. So we use the planescope and produce the accuracy from 40 to 53%. It means that the accuracy cannot be so high because the complexity of
Or Ibu Herawani. Yeah. Okay. Yeah.
Okay. So our research is about antioxidant activity for Berbasenia forbaceae. So in Bengkulu, Berbasenia forbaceae is one of the most abundant seaweeds, but until now is underutilized by local people. So my team will prove about the antioxidant activity from Berbasenia forbaceae, and the result show that Berbasenia forbaceae have antioxidant activity, but in intermediate and weakest category. So in future research, maybe we can extract the purer extract to increase the antioxidant activity of Berbasenia forbaceae. Thank you.
Thank you very much. This is Harvey. Now I'd like to invite Mr. Harb. Oh, sorry. I thought it's Lai, yeah? Thank you. Or Mr. Harbu.
It's me, Mr. Solita.
Yeah, please.
My audio. Okay, thank you very much for the session. We are very happy to share our study and our finding. And in this study, we talk about battery in the glacial period in Pleistocene. And based on our study, we can find the indication of the land bridge between Water and Nusa Penida, and also Nusa Penida between Lombok. And by this study, we can understand that there is no evidence of that direct island geography in this region between Bali, Nusa Penida, and also Lombok. And by this study also, we can know that thank you so much for your attention, Mr. Samita.
Okay, thank you, Mr. Harbu. Yeah, this is very interesting studies. Okay. Next, I will invite Miss Nabila Putri, please. Your time, yeah.
Yes.
Sorry. Have I give her opportunity to present, yeah? Sorry, I miss. I'm sorry, I'm very sorry. So I think
an important aspect of the sustainable management of fisheries resource. Food preference can be used to determine trophic structure and relationship, predict change in the food chain, or evaluate the development of a species in the ecosystem. In total, 231 fish samples, consisting 91 males and 140 females, range from 80.6 to 23.8 centimeter, were collected from fishermen in the Gunung Kidul coastal water from January to March 2023. Each fish sample was measured for its total length and body weight, then was dissected to determine the sex, measure the length and volume of the cut, and determine the diet composition. Data analysis include relative cut length, food composition, frequency of occurrence, index of preponderance, trophic level, food nice breadth, and food overlapping. The result shown that relative cut length was less than 1, which indicates that big ice cat was a carnivorous fish and does not show diet shift during their life. Similar result was shown by big ice cat in the Manowari coastal water. The food items found in the stomach of big ice cat consists of crustaceans, fish, squid, and detritus. Male big ice cat feed on the 4 types of food, where the female one only feed on 3 types of food. Crustaceans were the main food for big ice cat, with the complementary food was fish and additional food was squid. Almodar and Shueisha found a similar food type on the big ice cat in Oho Ililir waters, with feeds mainly on the crustacean. Based on the value of nest breed, male big ice cat are more varied in the feeding food than the female one. The male and female big ice cat showed intense competition in the feeding food with an overlap value of 0.935. Thank you very much.
Okay. I just wonder if Mr. Waskito is on the screen with us on the line.
Okay,
Okay.
I am the member of this.
Okay. Dr.
Sorry.
Setiabudi, please make a brief statement for this poster. Yeah.
Okay, thank you very much for the committee to the opportunity to participate in this event. This research regarding fishery biology conducted in the southern coast of Yogyakarta. Actually, there are many fish resources in this place. So I would like to know about the fishery biology aspect, such as reproduction and also food preference and so on. So in this research, I emphasis on the food preference of big ice cat or Sclarochromnophthalmus. I think it's important to completing the data and information as basis for fisheries resource management. And the results on that the crustacean is the main food of the black ice cat. Maybe in similar place also get a similar result. But as the most important is fish maybe adapt to the environment. The available food will change maybe diet safe on or other. And this research also known that there are so intense competition in feeding food in the male and female big ice cat. Maybe just this one I can explain about this research. Thank you very much.
Thank you, Dr. Setiabudi. Yeah. Thank you very much. Now I think I would like to invite Ms. Andini Nabila Putri, yeah, for a brief statement before we close this poster session on the second day of the symposium. Please.
Yes, I'm very honored and thank you to be able to take part in this presentation. And the poster that I upload is explained about the potential use of gelatin on surimi. And from this research, I know that scalefish from tilapia can use for the raw material for gelatin, and that has good yield and also gill strength. And the characteristic the gelatin from tilapia scale has appropriate with standard BSN. So and the application to surimi also has significant effect on the texture surimi gill that we want. So I think the gelatin in this research also have a next potential to application in other food. Maybe. Thank you.
Okay, thank you, Ms. Andini Putri. Okay, I just want to make sure. I need the operator or the committee to let me know. Is it the right time for just to make sure that all the presenters already given opportunity to briefly make a statement and we don't left any of the poster? Okay, thank you. Okay, ladies and gentlemen, I think we have a very good and very interesting poster presentation, yeah? All are very important. And if we connect to the speech of the keynote speakers, all of this content are very relevant, yeah, for developing Indonesia in terms of blue economy. I think it is very important, and the range of the content is very wide, yeah, from the study of the past, from biogeographical studies to the improvement of the food quality. For that, I would like to thank all the presenters, yeah, and to the audience for your attention following this session. So, without further ado, I would like to conclude this poster presentation. Thank you very much.
Okay, thank you, Mr. Fedis Wandita, for the smooth guidance through the poster presentation session. And to the next agenda, I think we will have a small break until 1 p.m., 13:00 sharp. So if you don't have any questions left, thank you for your attention up to now, and see you at 13:01 p.m. sharp.
And we would like to remind you to fill in the absence link that I've put it in the chat box below. You can also scan the QR link that is already seen on your own screen.
Okay, so if you have already filled the attendance link, I think it's time for us to take a break, a small break. See you at 1 p.m. sharp. Thank you.